The process of meiosis results in the formation of 4 daughter cells, each containing half the number of chromosomes found in the parent cell. In this case, the parent cell has 4 chromosomes, and the daughter cells have 2 chromosomes. The chromosomes are shown here as they look during cell division. In the parent cell, the chromosomes have replicated and condensed. Each chromosome consists of 2 sister chromatids joined at the centromere. The parent cell has 2 sets of chromosomes. One set of chromosomes, colored red here, came from the mother. The other set, colored in blue, came from the father. The matching pairs of chromosomes, one from each parent, are called homologous chromosomes. Homologous chromosomes carry genes for the same traits and are the same size and shape. A cell that contains 2 complete sets of chromosomes is called a diploid cell. The parent cell divides twice during meiosis. The first cell division is called meiosis I. During meiosis I, the homologous chromosomes are separate. 2 new cells are formed, each containing half the chromosomes from the parent cell. A cell with only 1 set of chromosomes is called a haploid cell. The second cell division is called meiosis II. During meiosis II, the sister chromatids of each chromosome separate. The 4 daughter cells are also haploid cells.
Imagine a diploid cell during interphase. The chromosomes are long, thin strands of DNA within the nuclear envelope. There is a single pair of centrioles near the nucleus. Near the end of interphase, the cell prepares for the first phase of meiosis, meiosis I. The centriole pair duplicates. Each chromosome is replicated. Homologous chromosomes begin to group together. Meiosis I begins. The first phase of meiosis I is prophase I. During prophase I, the centriole pairs move to opposite sides of the cell. The chromosomes condense. The homologous chromosomes are tangled together. A total of 4 chromatids are present, since each chromosome was replicated before meiosis began. Each pair of sister chromatids is joined at a centromere. While the pairs of chromosomes are close together, they may break and exchange segments. This is called crossing over. The nuclear envelope dissolves and the spindle fibers form. The homologous chromosomes remain paired as spindle fibers pull them toward the center of the cell. The second phase of meiosis I is metaphase I. During metaphase I, the homologous chromosomes line up in the center of the spindle. The third phase of meiosis I is anaphase I. During anaphase I, the homologous chromosomes are pulled apart by the spindle fibers. They move to opposite ends of the cell. The fourth phase of meiosis I is telophase I. In some cells, the nuclear envelope forms around the chromosome sets. Cytokinesis begins when the cell membrane starts to pinch the cell's cytoplasm in half. 2 new daughter cells are formed. Each daughter cell is haploid. Each has half the chromosomes from the parent cell. In preparation for another cell division, the centriole pairs duplicate. The chromosomes do not duplicate. Each daughter cell is now ready for meiosis II, the second cell division of meiosis. Meiosis II is very similar to mitosis. However, meiosis II starts with a haploid cell. The first phase of meiosis II is prophase II. During prophase II, the centriole pairs move to opposite ends of the cell. The nuclear envelope dissolves and a spindle forms. The chromosomes are pulled toward the middle of the cell. During metaphase 2, the second phase of meiosis II, the chromosomes line up in the center of the spindle. In anaphase 2, the centromeres of the sister chromatids separate. The chromatids are moved to opposite ends of the cell. Each sister chromatid is now considered an individual daughter chromosome. The last phase of meiosis II is telophase II. During telophase II, nuclear envelopes form around the chromosomes. In cytokinesis, the cell membrane pinches the cell in half. Each haploid cell has now divided into 2 haploid daughter cells. The original diploid cell has given rise to 4 daughter cells, each with a single set of unduplicated chromosomes. The chromosomes of all organisms contain regions called genes. Each gene determines a particular trait of the organism. For example, one gene in pea plants determines seed color. Another gene determines seed shape. A diploid cell has 2 matching sets of chromosomes. Matching pairs of chromosomes are called homologous chromosomes. Homologous chromosomes carry the same genes in the same locations. Since all diploid cells have homologous chromosomes, each cell carries 2 copies of each gene. However, the 2 copies are not always identical. The 2 copies of the gene may specify 2 different versions of the same trait, seed color for example. One version of the gene on one chromosome may specify green seeds, while the version on the other chromosome may specify yellow seeds. The different versions of a gene are called alleles. The cell also carries 2 different alleles that determine seed shape. One allele for wrinkled seeds and the other allele for smooth seeds. Now consider what happens in meiosis. The chromosomes replicate. In metaphase 1 of meiosis, homologous pairs of chromosomes move to the middle of the cell. In anaphase 1, the homologous chromosomes are separated and pulled to opposite ends of the cell. 2 haploid daughter cells are formed. When the homologous chromosomes were separated, the alleles on those chromosomes were also separated. The allele for yellow seeds is now in the right daughter cell, while the allele for green seeds is in the left daughter cell. This process of allele separation is called segregation. The alleles that determine seed shape have also segregated. The allele for wrinkled seeds is in the left daughter cell, while the allele for smooth seeds is in the right daughter cell. During prophase 1 of meiosis, homologous chromosomes come together as pairs. While lined up in this way, non-sister chromatids break at corresponding points. The chromatids then switch fragments, exchanging segments. This process is called crossing over. Crossing over allows chromosomes to exchange alleles. As a result, alleles are brought together in new combinations.